Okay, so greetings. Thank you for joining us today. This is the first of 4 Invasive Crayfish Collaborative webinars. This is an educational series meant to address invasive crayfish topics that have been identified to be of interest by collaborative participants. This series is being funded by the Great Lakes Restoration Initiative. Today's topic is life histories, impacts, and mapping of invasive crayfish, what we know and where we should go. Today's presenter is Dr. Eric Larson. He is an assistant professor in natural resources and environmental sciences at the University of Illinois Urbana-Champaign. He has worked on native crayfish management in the Ozark Plateau, Arkansas, and Missouri, the Pacific Northwest, and Upper Midwest. He's published 35 papers on crayfish covering life histories, trophic ecology, distribution, habitat associations, and field sampling methodology. He has a BS in fisheries resources from the University of Idaho, Moscow, MS in biology from the University of Arkansas, Fayetteville, a PhD in aquatic and fishery science from the University of Washington, Seattle. He also worked as a postdoctoral researcher at the University of Tennessee, Knoxville, and the Daniel P. H. Hather Center for Conservation and Research at the John G. Shedd Aquarium in Chicago. So the webinar, if you want to participate, ask a question, there is a chat box you can use. Everyone will be on mute throughout this presentation. So if you do have questions, ask those. We'll try to address them as we go through the presentation or at the end. This presentation is being recorded and then it'll be archived at a later date through the ICC website. And with that, I will let Eric do his thing.
Great, thanks, Craig. I'm going to share my screen now. Okay, thanks for attending the webinar. To kind of kick us off, I've kind of been asked to respond to a number of different sections identified in the needs assessment. And so I'm going to work through some of our background knowledge on crayfish life history, impacts of invasive crayfish, and then tools in trying to map the ranges or habitats vulnerable to crayfish invasions. And I think some other topics like control and eradication are going to be covered in subsequent webinars, but I'll make connections between, say life history and control where I can. So I'll kind of kick us off here talking about crayfish life history. What do we know about reproductive timing and age to maturity and fecundity and ways that can inform risk assessment as well as a management or control of crayfish.
Hey, Eric.
Yeah.
Your screen, it's in the notes section, the notes.
Okay.
Just letting you
know. Did that fix it?
Yeah, you're good. Thank you.
Okay, great. So I do want to say, I want to start out by emphasizing what we mean by life history. Sometimes people apply this term more broadly than I will here. But we think of the series of events or changes or biological phenomena exhibited by an organism from its inception to its death. And this is something that Stephen Stearns wrote a book in the early 90s, The Evolution of Life Histories, identified things like size at birth, age at maturity, age and size specific reproductive investments. These are the things we focus on as life histories. And that doesn't necessarily include other ecological or biological traits, things that could be morphology, diets, habitat use. Those tend to be things that aren't necessarily under the umbrella of life history, but you still might want to know about, obviously, for preventing or managing crayfish invasions. But life history traits, these kind of time to maturity or reproductive investment type traits have been useful both for invasive crayfish risk assessment, and there's interest in using life history knowledge to guide a management or control. So I'll give kind of one example here. I worked during my PhD on trying to predict both invasion and extinction risk of crayfish in the southeastern United States. We used ecological and life history traits for this work. And one of our best traits at predicting risk of invasion For invasive crayfish, what we're calling extra-regional invaders here, things that establish in places very far from their native range, was fecundity. Crayfish that have higher kind of egg numbers per clutch were more likely to have been observed to become these extra-regional invaders. And that's been consistent with work for other taxa like mollusks, and in other risk assessment tools for invasive crayfish, like the Notre Dame, the ND STARE tool that was developed kind of in the Great Lakes region. So in my kind of past traits-based risk assessment work for crayfish, I got a little concerned that in that past example, focused on crayfish in the southeastern United States, I had used life history data from sources like state crayfish guidebooks for Kentucky or Missouri that might report things like fecundity or egg size based on 1 or 2 total individuals, not based on actual kind of actual life history studies that had followed the population through a few years and had more data to estimate things like fecundity from. So I set out with Bob DiStefano, who's a biologist at the Missouri Department of Conservation and an undergraduate at the University of Missouri, to try to say, what's the state of our knowledge of crayfish life histories in North America, kind of a United States and Canadian focus on crayfish life histories. And so we had done a literature review to identify crayfish life history papers, what taxa were represented by life history knowledge, and what these papers included. And I would say that although this is North American focused, that's getting you the majority of the world's crayfish diversity, and the patterns we found are likely transferable to other regions. If you're trying to do risk assessment for maybe blacklisting of crayfish from Australia, what we found, and I'll kind of return to this, is probably going to be reliable for predicting do we know anything about the life history of species. And I'd also say, the study is from 2013, so some life history studies have been published since, but not very many, and we're actually kind of updating this work for a kind of crayfish conservation review paper that's in development right now. But as part of this literature review on crayfish life histories, we identified a total of 78 life history studies and 59 papers, and so that means some papers covered more than one species. And this represented life history studies for about 12% of the 357 crayfish species in the US and Canada. And so that's a big number, 357, because like freshwater mussels or freshwater fish, we have really high diversity, say in the southeastern US and in the Tennessee River, in Alabama, Georgia, Florida. And as part of the needs assessment, people identified wanting to know more about crayfish life history, potentially to inform management. So what I wanted to stress here is we publish this as a database with the paper. If you're looking for life history knowledge for a specific crayfish, this is a great resource. I just kind of want to identify the existence of this database, and I'll come back to it, as a place to look for what's available in life history information for a given species. So we looked at this kind of broken down by different genera of crayfish. Some are better represented for life history knowledge than others. The genus Orconectes, many of which have now been revised to the genus Faxonius, is better represented than some other species-rich genera like Procambarus or Cambarus. So we found 20 Orconectes crayfish had published life history studies, but only 6 Procambarus species did. And some of the biases here, terrestrial burrowing crayfish, cave crayfish, those things that are difficult to study were especially underrepresented for life history studies. Crayfish that are already invasive in North America were well represented. Our impactful invaders tend to have a good amount of published life history knowledge out there. So for example, the signal crayfish, this is a crayfish native to the west coast of the US that's invasive in California, Europe, Japan. If you wanted to know something about the life history of the signal crayfish, there's 7 published studies for that, and those are reported in our database. Red swamp crayfish, a popular aquaculture and harvested species in the southern US that's emerging as an invader in Michigan, Illinois, Wisconsin. Red swamp crayfish have 6 published life history studies. Rusty crayfish, maybe our kind of best known invasive from the Great Lakes Basin, from the upper Midwest. There's 4 published life history studies on that species. So if you were doing risk assessment for signal crayfish in the Great Lakes region, you could find information on the species. If you wanted to know something about red swamp crayfish life history for eradication, there are a good number of published studies. Many of those are not necessarily from more northern latitudes, like the Great Lakes Basin. And so we could maybe have some concerns about how a life history study from Louisiana might transfer to Michigan. But there is a decent amount of published life history data for some of these invaders. We kind of looked at the geographic distribution of life history knowledge. And maybe kind of unsurprisingly, lower diversity regions with fewer native crayfish species have better coverage of their life history information. So about 60% of native species in the Great Lakes Basin have a published life history study. Not complete coverage, but our crayfish in northern latitudes or on the more species poor west coast tend to be well covered. As we move to these really highly diverse, high species richness regions, our life history coverage drops off a lot. So if you're in the Tennessee River watershed, you've got about a 10% coverage of the 65 to 70 native crayfish species that occur there. And this is something I would maybe say that this could be a place where within continent invaders are going to come from in the future. Rusty crayfish has invaded the Great Lakes Basin from the Ohio, and potentially with climate change, we might expect to see more, northward invasions of things that are in Tennessee, in Kentucky, in Alabama, that have really, really poor coverage of basic life history information. And I would say that if you're trying to screen for invaders from other continents, you can probably expect similar patterns, say, from Australia. Where commercially valuable species and species that have already become invasive, things like yabby or red claw crayfish, are going to have some good life history knowledge available. But a lot of the kind of narrow range endemics, which may still be in things like the aquarium trade, are probably unstudied in a lot of cases. We also kind of broke down within these life history papers we found, what do people record as life history? And some of the most common things are reproductive season, reproductive form. A lot of our crayfish in North America cycle between an inactive and active reproductive form 1 and form 2 in males. These studies often report growth rate, and then things that get reported less frequently. Egg size is rarely reported. Time from egg extrusion to egg hatching isn't reported, and even fecundity, a trait that we find to be really important in risk assessment, was only reported in about 63% of these life history studies. So our coverage of traits that might be predictive of invasion risk, even within these life history papers, is kind of incomplete. The other thing we kind of flagged in this paper is that our life history knowledge isn't keeping pace with our taxonomic knowledge for crayfish in North America. On average, we've looked at kind of 4 crayfish species lists for North America through time, from Hobbs 1972 through Taylor et al. 2007. Over the last 30 or 40 years, we're describing about 3 new crayfish species in North America each year. Sometimes that's splitting off a new species out of a previous species based on some morphological or molecular data. Sometimes that's discovering a species that's entirely new to science. I've kind of shown on here a bottlebrush crayfish, Procambarus simmonsii, a large-bodied crayfish in the kind of Tennessee River watershed in North Alabama that had just been missed by science until just a few years ago. So we're still discovering new crayfish species in North America, and yet we publish less than 1 life history study for a new crayfish species every year. So this kind of natural history knowledge of these species doesn't necessarily keep pace with their discovery. And probably the most useful thing for this webinar, if you need to find life history information for a given crayfish species, we published our database with the paper. It's an Excel file. I checked the link just yesterday. The link's still good. You know, sometimes these online appendices for journals break or degrade over time. So species by species, we give the paper title and authors. And you know, if you're having a hard time tracking down a PDF, that's something that for a given species we'd be happy to share with you also. And so as a little bit of an aside here, I focused on life history, right? These reproductive and kind of growth type traits. But I think other things like physiological tolerances or habitat requirements are similarly poorly studied in similarly biased ways for other crayfish. So I've identified here a paper from Jacob Westhoff, who's a biologist in Missouri, and Amanda Rosenberger in the USGS co-op units, who looked at a global review of studies on freshwater crayfish temperature tolerances and preferences. And they found 56 studies that had done, say, laboratory kind of physiological trials of crayfish temperature tolerances. But these only covered about 6% of extant crayfish species worldwide. And I might say in comparison to our 12% coverage for life history data on North American crayfish, we might conclude that these temperature relationships that could be really valuable for predicting where a crayfish can establish or spread to are less well known than life history information for a lot of these crayfish. So can life history information inform management? Something's already established in your jurisdiction. Can you control or eradicate a crayfish? The kind of University of Wisconsin group, Jake van der Zanden, Brian Roth, now at Michigan State. Gretchen Hansen has worked on this now in Minnesota. In trying to control rusty crayfish in a small lake in northern Wisconsin, we know, for example, that juvenile or young of year crayfish recruit poorly to traps. You can't necessarily get these age zeros or even age ones out by baited trapping, but they're very sensitive to fish predation, right? So in that control effort, they opted to change fishing regulations. You could potentially stock more predatory fish to try to increase that predation pressure on the young of the year, where we know that things like baited trapping are really biased towards larger, more aggressive adult crayfish. So these researchers had combined both trapping removal and predatory fish management to try to target different kind of life history stages of the crayfish they were trying to remove. And that project didn't completely eradicate rusty crayfish from their lake, but they did greatly reduce rusty crayfish abundances in a lake in northern Wisconsin. Another example, the lab of Scott Bonar at the Arizona Cooperative Fish and Wildlife Research Unit has been working for a number of years on trying to control non-native virile crayfish in the southwestern U.S. If you're in the Colorado River Basin, you should have no native crayfish at all to the entire Colorado watershed. And things like virile crayfish have invaded there and had impacts on native fish. So Rogowski et al. Out of Scott Bonner's lab had kind of looked at trying to use life history information to optimize control of invasive crayfish. They did mark recapture work and backpack electrofishing for removals, and they kind of proposed that removal in autumn, when crayfish had in their system molted over to being reproductively active, but were still easy to catch before stream temperatures had gotten colder, was the best time to eradicate, to try to control virile crayfish populations. My sense is that they haven't succeeded in eradicating virile crayfish in any of their study sites, but this kind of makes sense for the life history of this species in their system in terms of using life history information to inform the timing of an eradication effort. So I'll kind of roll over here from life history to talking about what we know about invasive crayfish impacts. Greg, are there any questions I should kind of respond to at this point?
No, not at this point. You're good.
Okay, so I'm happy to keep going. If you write out questions, I can do those between sections or also at the end of the webinar too. So I'll kind of transition here to talking about our understanding of impacts of invasive crayfish. And you know, invasive crayfish have had their strongest and often best documented impacts in regions that historically lacked any native crayfish at all. I just mentioned that the Colorado River Basin is someplace that didn't have native crayfish. When crayfish show up there and native organisms are naive to this kind of large-bodied benthic generalist, you tend to see big impacts in those cases. So one example would be Crater Lake, this caldera lake in Oregon that had no crayfish in it, National Park Crater Lake. The signal crayfish on the west coast was accidentally introduced into Crater Lake, where it has really strong negative impacts on an endemic newt, the Mazama newt. Lots of amphibians fare poorly when crayfish invade, and signal crayfish also do things like destroy aquatic macrophytes. Lots of crayfish destroy macrophytes that can have impacts on say fish populations, but also kind of ecosystem processes in lakes. So if you add a non-native crayfish to an ecosystem that's never had crayfish, you see big effects. If you add non-native crayfish to a system with a native crayfish, you also see impacts or harm. Our invasives often displace native crayfish through a number of mechanisms. That can be competition, disease transmission, hybridization, differential kind of susceptibility to fish predators. So we've seen both extinctions and Endangered Species Act listing of native crayfish in the US because of impacts of invasive crayfish. What we know a little less well is in these systems where a native crayfish is replaced by a non-native crayfish, how different is the ecosystem role of that non-native to the native it replaced? Are these things kind of equivalent or functionally redundant, or does that invasive crayfish do really different things than the native crayfish that were already in an ecosystem like the Great Lakes? So I'm going to walk through what we know about functional differences between non-native and native crayfish from a number of different approaches to asking this question. That includes results of mesocosm and enclosure experiments, stable isotope studies looking at diet between native and non-native crayfish, patterns in abundance between native and non-native crayfish, and then kind of whole food web effects, again, often inferred by stable isotopes. Does a food web change after a crayfish invades? So most of what we know about invasive crayfish impacts is from things like laboratory experiments, mesocosm experiments, or enclosure experiments in the field. And about 5 years ago, I published with a couple of other co-authors a meta-analysis of the ecological impacts of non-native crayfish. And one of the things that we looked at was studies that contrasted native and non-native crayfish effects on things that include algae, macrophytes, snails, invertebrates, and fish. We found only 8 studies that had contrasted native and non-native crayfish impacts in enclosures or mesocosms. And there are a few more studies than that hadn't necessarily reported their statistics in ways that we could synthesize in a meta-analysis. But it's relatively more common for people to do enclosure studies that only add an invasive crayfish rather than doing a contrast between a native and non-native crayfish in kind of sympatry. And of these 8 studies that looked at a number of responses, we only found 3 effect sizes where there was a difference between native and non-native crayfish. 2 of those, the non-native crayfish had stronger effects on the ecosystem response. In 1, the native crayfish had a stronger effect. So the kind of evidence from mesocosm studies of functional differences between native and non-native crayfish is relatively weak. In the majority of these studies, there's no significant difference in effect size between a native and non-native crayfish when compared to each other in the same study. And the thing I would caveat about that is we haven't done as much of this as you might expect. We had 8 studies that had reported their statistics in a way we could use in a meta-analysis that had ever made this comparison anywhere. Europe, the upper Midwest, Japan, those tend to be the places we've best studied invasive crayfish. So there is still some need here to contrast community and whole ecosystem effects of native to non-native crayfish. And of those studies that exist, these mesocosm or enclosure studies have been criticized for being short duration, small spatial scale experiments that might not tell you as much as something that's under field conditions over longer durations, right? So this is the argument people have made for using things like stable isotopes to infer the function of an organism in its field setting over weeks or months or even years. So a number of studies have used stable isotopes to try to contrast dietary differences between native and non-native crayfish in sympatry. One of these was from the Arkansas Ozarks by Dan Magulik and a co-author that looked at a native and non-native crayfish. And if you're familiar with stable isotopes, we have a ratio of carbon-13 to carbon-12 on an x-axis that tends to capture what source of primary producers you're using at the base of the food web for your diet. Is this a autochthonous algae or a lochthonous leaf litter? And we have a ratio of nitrogen 15 to nitrogen 14 on a y-axis that tends to capture trophic position, how much of your diet is dependent on animal matter. And in this case, the native and non-native crayfish were basically sitting right on top of each other in the food web of these Ozark streams. They were feeding on the same diet items. And if you ran a kind of mixing model of their diets in the system, you get really high overlap. So that might suggest that these things are competing. That could be a mechanism for displacement of the native by the non-native, but functionally, they may not be doing very different things in this food web. And something I kind of flag here, that these are crayfish that are in the same genus. These were Orconectes, now Faxinias crayfish. They're relatively closely related. People have done this in other systems. This is an example from Finland, where the invasive signal crayfish from Western North America is being contrasted to the native noble crayfish in Europe, which is often displaced by signal crayfish because of disease. These are results of a stable isotope mixing model based on a biplot like the one I just showed that found really high overlap in diet between signal and noble crayfish. They're getting similar proportions of their diet from macroinvertebrates, from detritus, from macrophytes in comparison to each other. And so these are authors who kind of concluded that replacement of noble crayfish by signal crayfish in these Finnish lakes may not alter their food webs very much. What I kind of flag here, these are crayfish in the same family. They're in the family Astacidae. They're not in the same genus, but they're relatively closely related to each other in comparison to things that are in different families. So here's an example from my PhD research in urban lakes around Seattle, where we again use stable isotopes to look at trophic position and trophic overlap between the signal crayfish and red swamp crayfish that have invaded these Seattle area lakes from Louisiana. In this case, we found that signal crayfish were reliably higher in trophic position than red swamp crayfish, and that was ultimately true if they were in sympatry or allopatric, and it was supported by mesocosm work we did feeding these crayfish snails, Chinese mystery snails as well. And what I would kind of flag here is that these crayfish are in different families. Signal crayfish are in the family Astacidae, and red swamp crayfish are in the family Cambaridae. They're very distant from each other by their kind of evolutionary history. And something I've been thinking about but haven't tackled yet is, do phylogenetic or evolutionary relationships explain these differences? That crayfish that are congeners might be likely to have very similar diets. Crayfish that are distantly related in different families may be more likely to be functionally different from each other. And if that's true, something that I think we should investigate, then it might tell you that what crayfish that can invade the Great Lakes Basin is going to do something really different than the crayfish you already have. It might be something invading from a really different evolutionary background than the crayfish you have. Maybe apply more precautionary concern or criticism to things coming from Australia than from within a neighboring watershed. Another thing that's really important when we think about impacts of invasive species, the two kind of previous approaches, mesocosm experiments or stable isotopes, these may be things that are looking at per capita effects, individual effects of individual organisms. We have a number of ways of thinking about impacts of invasive species. This is kind of an old but famous formula from the late 90s about how we assess impact between different invaders. So these authors propose that impact is this kind of a formula that includes range, how widespread is something, What are its effects per individual, its kind of per capita effects, and how abundant is it? So we might expect a high impact invader to be something that's widespread, has strong effects as individuals, and is hyperabundant compared to native species. So maybe it doesn't matter if effects between individuals of native and non-native crayfish are similar, if non-native crayfish are reliably more abundant than natives. Impact may be about having a higher population size than the native species you've replaced. And people have looked at this for freshwater invaders in general, including invasive crayfish. So this is a paper from Gretchen Hansen and co-authors that came out a few years ago that was contrasting the kind of population abundance of native and invasive aquatic species. And they did find that aquatic invasive species are generally more abundant than natives, and this relationship was probably strongest for crayfish relative to all other taxa they considered. So the impacts we see of invasive crayfish may be about their abundance in comparison to the natives they've replaced. The thing I would caveat about this study is that this is based on two invasive crayfish. This is rusty crayfish in northern Wisconsin in comparison to native crayfish there, and signal crayfish on the west coast of North America, where they're invasive in northern California. So this is an N of 2. This is about 2 species. This hasn't been tested in a lot of other systems to characterize how generalizable this is across all invasive crayfish in comparison to their natives. So we actually do just need more studies that contrast non-native to native crayfish abundance, and studies that relate those differences in abundance to impacts to whole food webs. So I'll kind of wrap up here on impact by asking, what do we know about the food web effects of non-native versus native crayfish? These tend to be studies that have applied things like stable isotopes to ask how a food web changes pre and post biological invasion, or between invaded and uninvaded lakes. So for example, rusty crayfish in northern Wisconsin, going back to that Gretchen Hansen paper, we do know that rusty crayfish tend to be hyperabundant compared to the natives they've displaced. For example, here's a series of lakes, 5 or 6 lakes with rusty crayfish that have average catch per unit effort in traps of 25 to 35 rusty crayfish per trap, in comparison to the native virile crayfish that tends to be much less abundant in lakes that rusty crayfish haven't invaded. So we might expect that this hyperabundance of rusty crayfish changes food webs in ways that we could measure with stable isotopes. So our prediction here is that rusty crayfish have invaded these northern Wisconsin lakes. They're hyperabundant compared to native crayfish, and the things they do as they're hyperabundant is they destroy aquatic macrophytes, they reduce the populations of other benthic macroinvertebrates, things like snails, and they may potentially reduce populations of some small-bodied fish in these lakes, partly through destroying macrophytes that's shelter habitat from other fish predators. So the prediction we make on this gradient of high abundance rusty crayfish invasions is that they should increase the benthic food web dependence of predatory fish, that there's less use of the pelagic food web as small-bodied fish have been affected by macrophyte loss, that this is going to pull predatory fish towards the nearshore littoral benthic zone, and it should decrease the trophic position of predatory fish as they feed more on crayfish and less on small-bodied fish. So in a study that I had worked on with David Lodge and Tim Krebs, we found this generally to be true, but the effect to be weak. We looked at 6 fish species in lakes that have been invaded or not invaded by rusty crayfish. We found that for most fish species, their trophic position declined from white to black bars here with rusty crayfish invasions. That was only significant for walleye, though, although that's kind of interesting as kind of a very piscivorous fish in these lakes. All fish also increased their littoral dependence. They reduced their use of the pelagic food web and increased their use of the littoral food web with rusty crayfish invasions. And this was really strong for things like yellow perch that went from a relatively little use of the littoral food web in lakes without rusty crayfish to getting about half of their energy from the littoral food web when rusty crayfish had invaded. And so I note that this is consistent with another study in the same system out of the University of Wisconsin by Jake van der Zanden and students and postdocs, that used some of the same lakes in northern Wisconsin, applied kind of different quantitative tools on the stable isotope side, and similarly found that rusty crayfish had real but kind of subtle effects on whole lake food webs after they invaded. But to my knowledge, these are the two studies that exist that have contrasted native to non-native crayfish whole community or ecosystem effects, especially in field conditions with things like stable isotopes. And so this is something that we could use more work on in systems where these type of contrasts are possible, either before, after impact studies, or kind of a control impact of invaded and uninvaded lakes or streams. So should we worry about crayfish invasions transforming freshwater ecosystems in places that already had native crayfish? You know, for mesocosm and enclosure experiments, differences between native and non-native species have been relatively rare, but we could use more of these studies. This has not been looked at necessarily as much as studies that just compare invasive crayfish presence to no crayfish in a mesocosm. In terms of kind of contrasting diet between native and non-native crayfish with stable isotopes, sometimes we see no difference in diet between these kind of generalist omnivores. Sometimes we see really big differences. And I might propose that we should look at that kind of in the context of phylogenetic or evolutionary effects. Are things that are less related to each other more likely to differ functionally? We do probably have our strongest evidence that the impact of invasive crayfish is mediated by abundance. Invasive crayfish seem to be hyperabundant compared to the natives they replace, but that's based on observations from only two systems, northern Wisconsin and the UP of Michigan and California, and that's something that wouldn't be that hard to study, but not very many people have done or have published data in ways that's very easy to use. And then finally, you know, we do see significant but small effects of whole food web changes with crayfish invasions on this kind of hyperabundant gradient. But again, that's something we really only know from these well-studied lakes in northern Wisconsin. We've got a lot of knowledge gaps there. So I'll kind of roll over now into my last section on trying to predict where invasive crayfish might establish with kind of a case study of red swamp crayfish in the Great Lakes. At a glance, it looks like we don't have any questions, Greg.
We have one from Brian Roth.
Yeah.
So he's asking, do functional similarities might not equate to impact on the ecosystem, given differences in abundance that some crayfish attained? How is this accounted for in these studies? But I think he kind of answered it, so. So.
Yeah. Yeah, so you know, as we kind of dissect that, you know, per capita effects and the abundance piece, you know, those could interact in kind of interesting ways. So I'll kind of keep going into distribution modeling, and then we can do questions at the end. There should be maybe another 12 minutes or so. So something I've worked a fair amount on is trying to build species distribution or ecological niche models that extrapolate well between different geographic regions. And this is something that people working on these type of models have found to be challenging. That extrapolation to a new geographic range is a big challenge in using these type of models. And that might be because of niche shifts, that might be because of ways that the organism changes as a result of the invasion process. It could also be about just shortcomings of the model and trying to predict to non-analog climates or environments, and trying to predict to places outside the range of environmental conditions that a model was kind of tested and trained on. And this is something that's not always about methodology, about the choice of model or model algorithm you use, but something that I tend to think is more about incomplete representation of the fundamental niche. Every place a species can live by the type of limited presence-only occurrence data we often have available to use. So I'll give a kind of simple example of this, working with our red swamp crayfish, Procambarus clarkii, a species that a lot of people have concerns about in the Great Lakes Basin. During my PhD, I kind of looked at how the native range of the species compared to the non-native range and generating global species distribution models. So in my map here, we have dark red point occurrences that are in the native range of the red swamp crayfish, and light red points, we have non-native occurrences that I could georeference almost a decade ago now. And we know that we missed a lot there, that this is a species that's very widespread in China, where we couldn't just georeference locations at this time. But this was a pretty good coverage of the global range of this invader as of about 2011 or 2012. And I ran species distribution models based at these presence localities using a true presence-only species distribution model, something that doesn't require background or kind of pseudo-absence data. And I ran this just on terrestrial maximum and minimum temperature, the kind of temperature envelope of where this species occurs. So this is a pretty simple model, and it's a model that you would expect would overpredict where this animal could live in comparison to more complex models like maximum entropy that can really fit complex relationships to environmental data. So what this looks like, on the left, we have minimum temperature on an x-axis and maximum temperature on a y-axis for the native range and the non-native range, where warmer colors indicate places that are better for red swamp crayfish to establish. And the native range does a fairly poor job of predicting every place red swamp crayfish subsequently established as an invader. It doesn't predict much of Western Europe, where the species was very successful. It doesn't predict a lot of the West Coast, where the species is established. And it only predicts about half of the range in China where red swamp crayfish we know has established populations. And this tends to be based less on minimum temperature tolerances and more on maximum temperature tolerances. What we've kind of learned from this distribution model is that summers don't have to get as warm as the native range for red swamp crayfish for it to establish. It can tolerate places that are cooler in the summer than we might predict from the native range. And the other thing I would say about this model is neither the native or total kind of native plus non-native range model we ran here predicted most of the Great Lakes as a very good place for this species to establish. And we have reasons to suspect that's not true, right? This is a crayfish that is established in ponds near Lake Michigan and Wisconsin, in the North Branch of the Chicago River around Chicago. And it's had this big boom in Michigan that Brian Roth and others have worked on in lakes and ponds in say, the Detroit area. So we have known since about 1967 that red swamp crayfish have populations established in the kind of western basin of Lake Erie, particularly around kind of the Sandusky Bay region. So I think there's a lot of interest in understanding if red swamp crayfish are going to establish in more of the Great Lakes over time. Not just inland waters, but would we expect to see this crayfish in Great Lakes waters in the future? And it's been there for a while. It's been there, you know, for approaching 50 years. You may say, you know, if it was going to establish in Lake Ontario or Lake Huron, shouldn't that have happened already? The exception to that is that we know that time lags, long time lags between initial establishment and spread are fairly common for invasive species. And that's what we saw for rusty crayfish in the Great Lakes over time. Rusty crayfish has been known from the western basin of Lake Erie since the late 1800s, but it really didn't boom or spread in the Great Lakes until kind of the 1990s to 2000s. So this is a time series from Jody Peters' paper with a number of Great Lakes co-authors, following the spread of rusty crayfish from museum and agency records around the Great Lakes from the 1880s until the 2000s. Where rusty crayfish just kind of sat in a few locations in the Great Lakes for 100 years before they spread throughout the entire basin. And so something that's kind of I was curious about is, could we build distribution models that predicted every place rusty crayfish could live in the Great Lakes only from its earliest occurrences? Would we know from 1880 or even 1930 where that crayfish could live in the Great Lakes if we only had its earliest occurrence records? So a graduate student of mine, Rachel Egley, and I set out to try to see if we could build a species distribution model based on only those Western Lake Erie rusty crayfish occurrences that would predict the subsequent basin-wide invasion of that crayfish. And if we could, that would increase our confidence that we could do something similar for red swamp crayfish. Could we take this handful of occurrences from the western basin of Lake Erie and predict where red swamp crayfish might live in the Great Lakes 100 years in the future? So the way we tried to do this for rusty crayfish is we wanted to start from knowing a lot about the animal in the Great Lakes. We took its kind of a full occurrence data as of 2008. We built a species distribution model on the Great Lakes Aquatic Habitat Framework GIS layers. We identified which predictors best explained where rusty crayfish could live, and we took that kind of prior knowledge to go back in time and say, if we knew the future, could we build a good model for rusty crayfish? So we start, in kind of pre-1955 occurrences for rusty crayfish from Western Lake Erie. We only use the best predictors from the full occurrence model. We've kind of seen the future of where this animal is going to go. And we did a number of things to constrain the model to try to keep it from overfitting, to try to make it a little more generalizable to new areas in the Great Lakes. And even doing that, constraining the model complexity and knowing from the future what predictors seem to align well with rusty crayfish habitat, we could not build a good rusty crayfish species distribution model from its earliest occurrences. And so I plotted what that model predicts in the bottom panel here, the kind of C panel. Those Western Lake Erie occurrences say, sure, we think rusty crayfish are going to establish in Saginaw Bay, but nowhere else in the Great Lakes. That's what's most similar to Western Lake Erie in the ways that matter to this animal. So what we've kind of taken away from this is for something that's established with only a few populations in the Great Lakes, we can't use these kind of correlative SDMs to predict suitable habitat for that organism in the future. If we only have a few occurrences early in the invasion process, we're not going to be able to predict where this thing's going to live. And people have seen that in other systems and with kind of terrestrial invasive species, that early occurrence records in a new range do a bad job of predicting where something's going to spread to. So this is kind of frustrating if you're trying to anticipate, should I worry about red swamp crayfish in Lake Ontario? Should I worry about red swamp crayfish in Green Bay? Our kind of total global species distribution models at present, don't predict the Great Lakes as generally suitable for red swamp crayfish, and we can't really predict their future spread from their established populations in the Great Lakes. So is there anything we can do to try to identify where red swamp crayfish are going to live? So the approach we took in kind of facing these challenges was we set out to model crayfish habitat in the Great Lakes in general. Where do crayfish across all species live in the Great Lakes? And then can we use some of the known physiology of red swamp crayfish to constrain that model to the places where we think it can live? So we again kind of used this Peters et al. Great Lakes crayfish database published in the Journal of Great Lakes Research a few years ago, and we took the post-1990s crayfish occurrence records to build a habitat or a species distribution model. And we did that to kind of try to focus on a recent Great Lakes consistent with how the lakes have changed with zebra or quagga mussel invasions. We have some true absences, some places people looked for crayfish and didn't find them in the Great Lakes, but not enough to necessarily run a species distribution model on. So these models often use pseudo-absence or background data, where the algorithm's trying to differentiate true presences from the average background environmental conditions across the system to try to predict where something should live. And we again modeled this with a machine learning algorithm on the Great Lakes Aquatic Habitat Framework layers. And this kind of, the model fits the kind of known presences of crayfish in the Great Lakes really well. And I don't want to dwell too much on the model because it's just kind of one step of us trying to say, where should you worry about red swamp crayfish? But this is the model we built of what is crayfish habitat in the Great Lakes. If you're in our blue color ramp here, those are places we don't predict crayfish to occur in the Great Lakes. Once that flips at the threshold to the yellow-red color ramp, those are places we predict crayfish can live in the Great Lakes, with kind of increasing certainty as you move towards red. So this is kind of a nice map in terms of thinking about where you might find crayfish in the Great Lakes. We took this map and then we said, what do we think constrains from the physiology of the species where red swamp crayfish can live? And a study that I really like was run in Japan in the 1950s in kind of a cool temperate climate that looked at temperature triggers for red swamp crayfish reproduction. And we kind of chose summer temperature, maximum temperatures, as the physiological constraint for where red swamp crayfish can live, because there's pretty good recent evidence that cold temperatures don't kill red swamp crayfish, that we don't think minimum temperature is necessarily what's constraining where this organism establishes. So Veasley et al. Here from 2015 was a study in Eastern Europe that kept a number of semi-tropical or tropical crayfish outdoors in mesocosms. I think in the Czech Republic, I might have that wrong. A number of the Australian tropical crayfish really struggled. They died over winter. They didn't forage. But red swamp crayfish, Procambarus clarkii, at about 3 months at 3 to 4 degrees Celsius, the temperatures you might expect on a lake bottom under ice in the winter, not only had no mortality, but eventually, after a couple of months, decided to start foraging again, even at really low temps. So we have some skepticism that low temperatures necessarily kill red swamp crayfish, but instead, Suko 1958, working in Japan, and there's a series of English language papers that he published, found minimum temperature requirements for red swamp crayfish to initiate reproduction. So after a number of years of observing red swamp crayfish in outdoor ponds in the mountains of central Japan, they found a 15 degrees Celsius minimum temperature threshold for red swamp crayfish to start copulating and laying eggs. And reproduction peaked at temperatures of about 20 to 23 degrees Celsius. So we kind of took the benthic temperature model from the GLAF layers and clipped our Great Lakes crayfish model to the places we would predict would be warm enough for red swamp crayfish to attempt reproduction. So this is that kind of a crayfish habitat model for the Great Lakes in general, with that same color ramp. If something's in light gray, that's someplace we predict is too cold for red swamp crayfish to attempt reproduction. If something's in dark gray, that's someplace we don't have a benthic temperature prediction from GLAF. So unfortunately, that excludes Lake St. Clair or Sandusky Bay, places that we probably think red swamp crayfish could do pretty well, but it does capture a lot of the rest of the Great Lakes. If we make that clip at a 15 degrees Celsius cutoff based on Suko, we would predict 2% of the Great Lakes as suitable for red swamp crayfish. If we increase that to 20 degrees Celsius, where's an optimal place for this crayfish to reproduce, we predict about 0.9% of the Great Lakes as suitable. And that kicks out all of Lake Superior as too cold. But if you're in Lake Ontario, Lake Erie, Lake Huron, Lake Michigan, there are nearshore locations with good crayfish habitat that we think are warm enough for red swamp crayfish to establish populations. And so here's a couple of examples. We would predict Saginaw Bay to potentially have a lot of places that could be invaded by red swamp crayfish. The western basin of Lake Erie, where we have seen red swamp crayfish in Sandusky Bay. Some of the southern Lake Michigan shoreline off of Chicago. And for example, a lot of Green Bay in Wisconsin or the Escanaba area in the Upper Peninsula of Michigan. We would predict as a place you might want to try to keep red swamp crayfish out of trade and to potentially monitor for establishment of populations as locations in the Great Lakes themselves this animal might establish. So the kind of good here, we do identify our best effort at priority areas where we think red swamp crayfish could establish in the Great Lakes, and that might guide regulatory enforcement, education and outreach, surveillance and monitoring. We've run environmental DNA for invasive crayfish in the Great Lakes, looking for rusty crayfish, and been able to detect crayfish successfully in the Great Lakes with eDNA. So this might identify places you would monitor for red swamp crayfish populations. The things we would caution about this model, the Great Lakes crayfish habitat model, the SDM is dominated by Orconectes or Faxinaeus species. And so things like rusty crayfish may have really different habitat needs, things like substrate than red swamp crayfish will. So if red swamp crayfish habitat requirements are really different than our model predicts, that's biased a little more towards these Orconectes species, we could misrepresent where this would live. And so what we kind of want to know, how important is a preference for soft or silty substrates for the species for burrowing in the Great Lakes? And if that's kind of your expectation of where this animal will live, you could certainly use GLAF layers to say, not only what temperatures, benthic temperatures will this species reproduce, but where the substrates that it's likely to establish in. And I think, a number of mesocosm or lab experiments could help narrow these predictions farther. I think Ruben Keller at Loyola has been working on, kind of red swamp crayfish biotic interactions with other crayfish. Why haven't we seen the species established in Lake Michigan itself out of the North Branch Chicago River? And that could be because it's losing to crayfish already in the system, like rusty crayfish, or it may be vulnerable to fish predators. And the other thing I want to drop in here is that we can kind of do this because physiological data and temperature tolerance data exists for the red swamp crayfish. It doesn't for 94% of global crayfish species. So, we're going to struggle some in doing this for crayfish you might want to try to conduct risk assessment for, that these type of studies like Suko from Japan on red swamp don't exist in a lot of cases. So we're kind of coming up on being out of time this hour. The webinar series will continue on other topics in the future, potentially including some emphasis on control or eradication. And if we've got a little time, I'm happy to answer questions for as long as people want to stay on.
So there's one question. Have you tried using this model in Great Lakes Basin waters that are not the Great Lakes themselves, i.e. inland streams and lakes? It seems to the responder that a lot of Great Lakes crayfish don't live in the Great Lakes, but are abundant inland.
Yeah, so I intend to. There's a number of good and increasingly good inland freshwater GIS layers that could be used for this. We've kind of focused on this study in the Great Lakes themselves, but I think SDM using, for example, Sami Domich and Walter Yatz at Yale published a global 1 kilometer resolution freshwater habitat database. It includes for every 1 kilometer freshwater grid cell, streams, rivers, lakes globally, precipitation, upstream temperature, upstream land use, that in the past, many of us have used terrestrial climate surrogates and SDMs for freshwater distribution modeling and risk assessment. Now that we have kind of a freshwater version of something like WorldClim, we should be using that. And that there are data sets available that would be really nice to take these models inland in the Great Lakes Basin.
Okay, another question is, can you summarize where you think we should focus research efforts in terms of application to management?
Yeah, I really, I think crayfish are really challenging to manage once established, and they're hard to eradicate. Their spread can be difficult to control because of their ability to spread overland around dispersal barriers. And I think my priority would be identifying species at risk of becoming invasive in the future, approaching preventing those from being introduced to the Great Lakes Basin, either on kind of the policy side or an outreach side to users of organisms in trade. I've worked on invasive crayfish control. I've worked on systems where barriers have been built to try to contain the spread of crayfish once introduced. And I'm probably more skeptical than some people who will participate in the webinar series about the outcomes of management once something's in your system. I would direct the majority of time and resources to trying to prevent new things from being introduced into the Great Lakes.
Okay, I'm not seeing any more questions. So I guess we could probably wrap it up. And there's always, you could contact us again for more questions for Eric from this webinar.
Yeah, and if you'd like a copy of any of the papers covered or the life history database that I touched on briefly, if you send me an email, I'm happy to send stuff along.
Okay, great. And we will be archiving this webinar on our Invasive Crayfish Collaborative website once that is launched. And then, yeah, that's about it for today. Look out for other future webinars. And we can also send out an email with a bunch of the papers to the ICC. So, okay. All right. Well, thank you very much for your time, Eric. It was really great. And thank you, everyone, for participating.
Right. Thanks.
All right. Bye.